Hi friends. So let's start discussing bio mini test 5. This is the official scheme this time for this paper. We wanted to do something different this time. Okay, so let's start discussing. You have 15 questions and you have been given 30 minutes and you have to make the best out of those 30 minutes to read the questions carefully and understand the approach these questions. That's the reason why we gave 30 minutes. Most of y'all make mistakes because y'all have not understood the question properly, or you get confused with the options and having no confidence in your response. So let's try to build confidence through this paper, experience through this paper too. Okay, let's start with question number 1. With regards to eukaryotes, now you have to select which of the following is correct. The first statement says both 70S and 80S ribosomes exist as free and bound ribosomes. Now this is incorrect because the 80S ribosomes may exist as free or bound, like they may exist freely in the cytosol of the living cell or as ribosomes or polyribosomes, or else they may remain bound on the outer membrane of nuclear envelope or the rough endoplasmic reticulum. So 80S ribosomes, yes, they may exist as free or bound, but 70S ribosomes, they exist only freely in the stroma of chloroplast and matrix of mitochondria. They are present in eukaryotes, but only freely in this particular gelatinous material within that organelle. So option 1 is incorrect. Then B, initiator amino acid is formyl methionine. You know very well that domain eukarya, their initiator amino acid is for protein synthesis is methionine, and also in domain archaea it is methionine. Domain bacteria, the initiator amino acid is formyl methionine. Then option C, flagella are present as an extracellular structure. No, eukaryotes, their flagella is intracellular, while bacteria like prokaryotes, their flagella is extracellular. So therefore option C is also incorrect. And you know some other stuff about the flagella, it is intracellular, enclosed within the plasma membrane. That's why we say it's intracellular, and it has a 9+2 arrangement of microtubules, as you know, and also the average diameter is around 200 nanometers. Option 4, oldest fossils of multicellular eukaryotes date back to 1.2 billion years. This is correct because the first eukaryotes fossils date back to 1.8 billion years ago. That's when the first eukaryotes evolved from the prokaryotes, and they were unicellular, and it's explained in the endosymbiotic theory. But over time, the multicellular eukaryotes developed, and it's mentioned in the resource 1.2 billion years ago. The oldest protists, the red algae, Ediacaran, they appeared. It's not mentioned so distinctly, but you'll know that the other 4 options are incorrect. So this is the most appropriate answer. And the fifth statement, all are chemoheterotrophs. No, now when you consider eukaryotes, the domain eukarya, it has kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia. If you take kingdom Plantae, their major mode of nutrition is photoautotrophic, while there are those who exhibit special modes of nutrition like carnivorous, insectivorous plants, and then there are those who involve symbiosis. Then if you take kingdom Animalia, they are all heterotrophic mode of nutrition, while there are also those who undergo symbiosis. Protists, they may be heterotrophs or photoautotrophs, and there are also mixotrophs like Euglena. Depending on the availability of light, they may be either photoautotrophs or heterotrophs. While fungi, they are heterotrophic absorptive mode of nutrition. So all being chemoheterotrophs is incorrect. Then option is 4. Again, oldest fossils of multicellular eukaryotes dates back to 1.2 billion years ago, and that was red algae called Ediacaran. That is just for an extra knowledge. Don't confuse with the oldest fossils of unicellular eukaryotes being 1.8 billion years ago. Okay, and so that is question number 1. Question number 2, which of the following is incorrect regarding animal cells? Now this question, let's observe the correct statements first, and then we'll come to why option 3 is incorrect. Option 1, nucleus houses chromosomal DNA and is surrounded by a double membrane bounded nuclear envelope. This is correct because I didn't say the nucleus houses chromosomes. I said chromosomal DNA. Now, in the case of prokaryotes, their main genetic material is found concentrated in the nuclear region, while they also have fragments of genetic material called plasmids elsewhere in the cytosol of that cell. But eukaryotes, like animal cells, their entire genetic material is housed within the nucleus, and their genetic material is the genetic material of the chromosome. That's why it's called chromosomal DNA. So it is housed within the nucleus, and the nucleus is composed of a double membrane structure, you know that. And between the outer and inner membranes, there's an intercellular space. So option 1 is correct. Option 2, endoplasmic reticulum is a system of membrane bounded compartments separated from the cytosol. This is correct because endoplasmic reticulum, you have rough and smooth endoplasmic reticulum. Rough endoplasmic reticulum is a stack of flattened fluid filled sacs stacked on top of the other one, like extending from the outer membrane of the nuclear envelope, while the smooth endoplasmic reticulum is a stack of fluid filled sacs, and they are membrane bounded. That's why they are considered as organelles, and they have an ER lumen inside. So they separate this lumen from the cytosol, from whatever the components of the cytosol and all. So that way option 2 is correct. Let's check option 4. Mitochondria are sites of oxidative respiration and ATP synthesis. When I talk about oxidative respiration, we are talking about also oxidative phosphorylation process and all, because oxidative respiration involves complete oxidation of the respiratory substrate. So it is the site of oxidative respiration, and during the process of cellular respiration, aerobic respiration, ATP synthesis occurs in the cytosol of the cell and also inside the matrix of mitochondria. So you can also consider it as the site of ATP synthesis by oxidative phosphorylation mainly, and also by substrate level phosphorylation also. While so this way this statement is also correct. It is the site of oxidative respiration because when you talk about complete oxidation during aerobic respiration, it involves the complete oxidation of the respiratory substrate, whereas during fermentation only incomplete oxidation occurs, and for complete oxidation they must enter the mitochondria. So option 4 is correct. And option 5, Golgi complex is a stack of flattened sacs and vesicles that collect and package substances made in the cell. This is correct. This is one of the main functions of Golgi. It collection, packaging and distribution of materials. It is one of the main functions. So distribution of substances and materials means it may be to transport elsewhere within the cell or to secrete outside of the cell also, but it is one of the main functions of Golgi. So that way, let's come to option 3. Most organelles of animal cells are visible with common light microscope. This statement is incorrect accordingly because the other statements are most appropriately correct. This is the most inappropriate one because if you check, but again there are some confusions with this question. This is the only question where we wanted to emphasize a point with the past paper. 2018 they mentioned that mitochondria is not visible under common light microscope, but we also know that if you study the latest edition of Campbell, they mentioned that mitochondria is between 7 to 10 micrometers. I guess, I'm not so sure, it was 1 to 7, I guess. 1 to 7 micrometers, the average size. So, and the resolving power of the common light microscope is 0.2 micrometers, so you definitely can see mitochondria too, but they mentioned that mitochondria is not visible. And other organelles like nucleus is 5 to 10 micrometers in diameter average, so you can see it under common light microscope. Basically the other organelles also to a certain extent may be visible, but most can be considered as not visible. We are not sure because they are not provided much information for us, but the other 4 options are correct, so you can go with option 3. Question 3, identify the correct sequence in ascending order for the above occurrence of events as per evidence by fossil records. Now this question, remember the resource book has given each of these statements in the correct order of the event taking place in the resource book. It's given as so, but they have not mentioned the dates. I will mention the dates also now. Now, thing is, first seed plants. The resource book makes another printing mistake over there, where as they keep talking about as they go from the oldest events that took place to the most recent events, they will go from 700 million years ago to 500 million years ago to so on. But in the middle, they mention all of a sudden 40 million years ago, vascular plants differentiated, roots, stem, and leaves in plants. They diversified such plants. It's actually supposed to be 400 million years ago, not 40 million years ago, because according to Campbell also, early vascular plants, that means having roots, stem, and leaves, they appeared between 444 to 416 million years ago, which is actually around 400 million years ago. So it's a small error in the resource book, but you have to go along with it. So first seed plants, however, appeared in when you consider the Phanerozoic eon, it appeared in the Paleozoic era around 359 to 299 million years ago. Then B, appearance of cnidarians. Cnidarians appeared actually in the Proterozoic eon. They appeared in the Proterozoic eon, and that was before you call it the Precambrian period, before the Cambrian explosion occurred, around 635 to 542 million years ago, along with other soft-bodied invertebrates like the mollusks and sponges. Cnidarians too appeared. Now remember these dates. Then evolution of aerobes. Aerobes evolved. There were aerobes back then too, but evolution of aerobes, evolving, better adapting to aerobic mode of respiration. This all happened after the evolution of oxygen generating photosynthesis in cyanobacteria, because that way oxygen concentration increased. So now they have ample amount of oxygen availability, and therefore they too evolved. So this took place around 2.7 billion years ago. D, appearance of multicellular eukaryotes. Multicellular eukaryotes, as we discussed before, took place around 1.2 billion years ago. And then E, origin of most present day insect groups. This too is mentioned as the last point under the Paleozoic era, and this took place around 299 to 251 million years ago. Most present day insect groups appeared in this period, while certain insects were also there in between 416 to 359 million years ago also. But accordingly, this is the correct answer. Then if you put them in the order of the oldest to most recent events, you will get evolution of aerobes first, then appearance of multicellular eukaryotes next, then appearance of cnidarians, and then first seed plants, and origin of most present day insect groups. So answer is C, D, B, A, E. Also, you can logically analyze almost the correct answer, because first, anyway, cnidarians, seed plants, they are all eukaryotes, insects all eukaryotes. So if they had to appear, first of all, multicellular eukaryotes should appear. Then D should be coming early on. But then multicellular eukaryotes are mainly aerobic. Even though they may have anaerobic modes of respiration, they are mainly aerobic too. Therefore, aerobic respiration must have evolved before. Therefore, C will come first and D comes next. Then what comes after is between B and A you have to choose. Now, thing is, cnidarians, it's like this. Cnidarians, they were in aquatic bodies, mainly marine but also freshwater. At that time, there were only mainly aquatic plants. So first seed plants appeared when plants started to colonize land, and not just colonize but vastly colonize land. Seed plants, they evolved seeds which helped them to transfer gametes in the absence of water. So now they could even colonize dry habitats. So seed plants evolved later on, way after cnidarians appeared. So cnidarians were there back then in the aquatic bodies. Then you can put B. Again, you may get confused with the thing that insect groups. Like I said before, now we know arthropods were also among the earliest animals to colonize land, around 470 million years ago. 450, 500 million years ago, you can say. But that doesn't mean all arthropods. There were certain arthropods, but insects are also arthropods. But most present day insect groups, this actually appeared in the Paleozoic era. This is according to the resource book. So that way option E is the last. But if we mention insect groups in common, remember that certain arthropods were there at the early stages of Paleozoic era too, and they colonized land along with the early plants and algae, fungi and all. So the correct answer is 2. Let's move to question 4. Which of the following are correct with respect to Nephrolepis? Nephrolepis is actually a phylum Pterophyta, and these guys, they have a dominant sporophytic generation. So dominant sporophytic generation, but in Nephrolepis their gametophytic generation also was independent. That's as mentioned in the resource book. It is independent, but thing is, it is not completely independent, because once sporophytic generation dominates, the other generation automatically becomes at least partially dependent on the higher generation. But gametophytic plant body also has photosynthetic tissues, and it is independent to a certain extent, while also dependent on a certain extent to the sporophytic generation. While also the sporophytic generation, until it develops its photosynthetic tissues, it will be dependent on the gametophytic generation. After that onwards, it is completely independent. So both of them are like partially independent, but then the statement D says independent sporophyte and gametophyte, so you can consider it as correct, because I never said completely independent. Then if you check option C, biflagellated sperms. Biflagellated sperms are found mostly in the Pogonatum and the Selaginella, spike mosses, not in Nephrolepis. These pterophytes like Nephrolepis, they have multiflagellated sperms. So C is incorrect. B, unisexual gametophyte. No, Nephrolepis, they have a bisexual gametophyte, and the gametophytic tissue, that plant body, it has both the antheridia and archegonia embedded on the ventral surface of that. You call that structure as a prothalloid body. So that's also incorrect. And then A is homosporous. Yes, Nephrolepis, they are homosporous in their dominant sporophytic generation. In the ventral surface, at the borders of the leaf, you have these clusters of these spore producing structures in clustered. The spore producing structures are called sporangia. They're clustered together into sori enclosed by indusium. So there are many sori, and in each sori there are clusters of sporangium, but all of these sporangium produce only one distinct type of spore by meiosis. So since only one type of spore is produced, it's called homosporous, and that will give rise to only one type of gametophyte. So option A and D is correct, then answer is A and D only. And also, to not make the question too difficult, we did not give you confusing other MCQ points around it also. So we made it a bit easy, but you can get experience from these questions. Question 5. All arthropods. Now this you have to be careful when you say all. First statement, have a closed circulatory system. No, all arthropods have an open circulatory system. But then again, you say insects usually do not have blood. It's okay. All of them have generally an open circulatory system, not a closed. So that statement is incorrect. Second one, are motile. Not all arthropods are motile. Generally, most are motile, but some are sessile, like the barnacles that are found embedded on the surface of the whales. They are sessile forms, but they get their nutrition and all as This is a type of symbiotic commensalism, you call this, because they embedded on the whales, and as the whales swim from location to location, they also are moved automatically from one place to another, and they can get the nutrition they want. But they are sessile actually. Even though it's not mentioned in the resource, you will find a more appropriate answer is correct, and that's why you can eliminate this point. Option 3, respire using book lungs. Well, book lungs is found only in the arachnids of arthropods. Arachnids means the scorpions and the spiders like species. So this is also incorrect. We'll check have hemolymph. Option 4, have hemolymph as circulatory fluid and interstitial fluid. Yes. Now usually in those who have closed circulatory system, they have one type of fluid enclosed within the vessels and another fluid which is surrounding the body tissues and all. That is called the interstitial fluid, and the one that is restricted within the vessels, interconnected vessels, is called as the circulatory fluid. But arthropods, they have an open circulatory system, which means they do not have a distinction between a circulatory fluid and interstitial fluid. It is just one common fluid which flows through the heart and which bathes all the organs and tissues of the body, and that is called the hemolymph. So that is correct. And if you check option 5, excrete using Malpighian tubules, this is incorrect because the crustaceans they excrete through green gland, or you can call it maxillary antennary gland. They excrete through that. So most have Malpighian tubules, but not all. So answer is answer 4. Question number 6. Which of the following polysaccharide function relationship is incorrect? You have to select the incorrect one. First one, glycogen stored in cytoplasm of animal cells. Now this is correct because glycogen, if you know, it is found as a storage polysaccharide in both animal cells and fungi, and it is actually stored in the cytoplasm, no one else. They don't have a cell wall, and it's stored in the cytoplasm of animal cells. Option B, cellulose component of algal cell wall. As mentioned in unit 2, cellulose is found as a component of both plant and algal cell wall, and the best evidence is ulva has cellulose mainly as their cell wall component, while other algae, they usually have other components also, like for example jellydium will have carrageenan also, but carrageenan is like the main component of the cell wall. But anyway, cellulose is found in the component of as a component of algal cell wall. Option 3, peptidoglycan as a component of archaebacterial cell wall. This is incorrect because peptidoglycan is found only in the cell wall of bacteria, domain bacteria. Only they have peptidoglycan. As you study in unit 3, domain eukarya, domain archaea and eukarya, they do not have peptidoglycan. They have only proteins and polysaccharides in their cell wall. So option 3 is incorrect. Option 4, inulin stored in tubers of artichokes. Inulin is usually as a storage polysaccharide, and it is stored in tubers usually, like root tubers of dahlia, tubers of artichokes. This is not mentioned as an example in the resource book, but 3 is a more appropriate answer. Fourth one, chitin component present in the cuticles of insects. Friends, please do not confuse between cuticles and exoskeleton. It is both the same thing. It is both an extracellular structure. And thing is, the cuticles of insects, it functions as an exoskeleton because you know arthropods, they undergo a process called ecdysis, a molting process. And the importance of ecdysis, you know, right? That is, now ecdysis, the reason why they undergo this molting process is that cuticle will actually pose a limitation to that continuous growth. So to overcome that limitation, only they shed that cuticle and they will replace the new one, and during that time they can grow and grow bigger and bigger. That is why they undergo ecdysis. Now it usually refers to shedding of the cuticle. Now arthropods, you know very well that chitin is stored in their exoskeleton, and exoskeleton and cuticle is the same thing actually, that is present outside as an extracellular structure. You cannot have both actually, and it is the same thing. Then option 3 is the correct one. Question number 7. Which of the following kingdoms consists of species which represent all the above characteristics? Here, what we are going to do is we'll use the method of elimination. See option C, formal methionine as initiator amino acid for protein synthesis. Formal methionine is found only in domain bacteria. So you can remove kingdom fungi, kingdom animalia, kingdom protista. You have only kingdom bacteria and kingdom cyanobacteria left. Between these two, both of them have formal methionine as the initiator amino acid. But then if you consider statement B, flagella as locomotive structure, know that most or generally cyanobacteria, they do not have flagella. Then option should be B, kingdom bacteria. And if you check statement A, cell wall consisting of polysaccharides, yes, peptidoglycan is also a polysaccharide. Therefore, kingdom bacteria has all 3 correct characteristics. Then options for question number 7 would be kingdom bacteria. second option. Question 8. Consider the following statements regarding invertebrates and select the correct statement. This question is more of a logical question which tests your general knowledge also, which we want to emphasize. Do not always by heart the resource book. You should also have your thinking capacity. So check statement A. Some arthropods have eyes on stalks. Yes, if you observe the crustaceans, they have eyes on stalks. So you can take that as correct because I said some arthropods. Then B. Ecdysis is observed only among species of arthropods. Again, no, I said ecdysis usually is observed in those who have cuticles. Nematodes have a cuticle and they also usually undergo ecdysis. It's a flexible but a tough cuticle, and they also undergo ecdysis. So it's not only observed among species of arthropods. So B is incorrect. C. All echinoderms have tube feet. Now a past paper point also says that this is correct, that all echinoderms have tube feet. So that way we'll move on with it as correct, because the resource book has not emphasized it. It has just simply said as a common characteristic found in echinoderms. And D. Mantle is found only in certain mollusks. Now mantle is actually one of the three main structures present in mollusks. It is a fold of tissue that drapes over the viscera, and its main function of the mantle is to secrete the shell as a protective layer. Now mostly the shell secreted will be an extracellular cell, externally found, like in the land snails, while some also have internal shells like the squids. But there are those who don't have shells also, like octopus, but they do have a mantle. Now the purpose of mantle is to secrete the shell, right? Usually those who do not have shells, then it does not necessarily need a mantle, but an exception I gave now is octopus. So mantle is not found in all, it is found in certain mollusks. So that statement can be taken as correct. So option would be A, C and D. If you had an option called A and D, then it would have been a bit controversial. So we have eliminated such a problem. Answer is A, C and D only. Question 9. Which of the following does not include as part of the apoplastic route? Now you know the apoplastic route refers to everything outside of the plasma membrane of the living cell. We are saying living cell. And when you say plasma membrane, you know the living cell has something called the protoplast. The protoplast consists of the entire living content of the cell, and the boundary of the protoplast is the plasma membrane. Everything outside of the plasma membrane that way becomes the part of the apoplastic route. It includes the cell wall, the intercellular or you can say extracellular spaces, and something called substomatal air cavity is also given in this question number 9. Option 5 here says substomatal air cavity. Substomatal air cavity is also an intercellular space, but the difference is intercellular spaces means usually between cells. That's why you say intercellular space. But substomatal air cavity is a type of extracellular space which is involved in contact between cells and atmosphere. Intercellular space means cells and cells, while substomatal air cavity means cells and atmosphere. That is where diffusion takes place during transpiration, loss of water. So it is also part of the apoplastic route. Intercellular space is correct, cell wall is correct, and also the entire interior of dead cells like tracheids and vessel elements also become part of the apoplastic route. The reason is the dead cells, their protoplast is dead, so that's why you can say it becomes part of the apoplastic route. So option 4, lumen of tracheids, is also correct, while option 2, lumen of sieve tube elements, it's incorrect because sieve tube elements are living cells. They do have lumen, and usually we say that their cytoplasm is also peripherally bound as a layer to the plasma membrane. That is so that there's more free space for conduction of the phloem sap. But then again, it's a living cell, and according to definition, it is not part of the apoplastic route. So answer is 2. Question 10. Which of the following plant growth regulators does not have a role to play with regards to seed germination? When I say does not have a role to play, it refers to maybe it's not involved in promotion or stimulation or inhibition, whatever. If it should not have anything to do with seed germination. Now, according to the teacher's guide, they have given that jasmonic acid promotes seed germination. So option B is correct. Option C, gibberellin it stimulates seed germination. Abscisic acid, option D, it actually inhibits seed germination during early stages of seed dormancy. While promoting seed dormancy, it inhibits early seed germination. And option 5, cytokinin. Yes, cytokinin also stimulates seed germination. Then if you come to option A, ethylene, this is incorrect because ethylene has nothing to do with seed germination, and the resource also has given you only a certain amount of functions like promote ripening of many fruits, promote flowering in the pineapple family, then enhance the rate of senescence, promote leaf abscission, promote root and root hair formation, and also promote the triple response in seedlings. So it has nothing to do with seed germination, so option is 1. Question 11. Which of the following protists show all above characteristics? Again, we are going to use the method of elimination here. Containing cell wall. See, option C says containing cell wall. From this, paramecium is a protist. And now in this question, another thing you can do. Now they said which are the following protists, right? So you should analyze and check whether all answers are protists. And here definitely all 5 options are protists. Then you go to a method of elimination. Containing cell wall. Option C says containing cell wall. Paramecium, they do not have cell wall. They have pellicle beneath their plasma membrane, but no cell wall. And euglena also no cell wall, they have only a pellicle, so you can remove option A and D. Then you have only gelidium, diatom and ulva. Now this one you can use option A or B because both gelidium and ulva live only in marine habitats. And if you consider statement B, that it says unicellular, both gelidium and ulva are multicellular. Okay, while again both option A and B will prove that only diatom is unicellular. Yes, and they exist as phytoplanktons and they live in both freshwater and marine habitats. That's correct. They are as one of the main primary producers in aquatic bodies like oceans because they exist as phytoplanktons. They're called the pastures of the sea. And yes, they do have cell wall. It's composed mainly of silica. So all above characteristics is shown by diatoms only. Then option for question number 11 is C, diatom. Question number 12 is the most popular question because at the time of recording we have 108 responses, alhamdulillah. From these 108 responses, only 14 students managed to get it correct, and I don't know how many of them understood the correct thing behind this question, but congrats to them. And others, I hope you all learned something from this. You have to read the question itself very carefully. Which of the following structural modifications of the dicot leaf? Now we have to focus to the structural modifications of the leaf, not the soil, not the plant body, nothing. We are going to focus on structural modifications of the dicot leaf. Cannot be considered as an adaptation to maximize light capture. Okay, first let's check whether all 5 options are involved in maximizing light capture. Transparency of cuticles. Leaves have cuticles, yes, and the transparency of cuticles actually enable more light to penetrate into the palisade cilia and all. So therefore it actually is an adaptation to maximize light capture. Yes, correct. Option 2, presence of palisade cilia perpendicular to epidermis. Now if you take the leaf, dicot leaf, usually the palisade cilia is found only on the upper epidermis, right? And the cells are elongated column shaped cells. They are usually packed perpendicular to the epidermal cells. Now the purpose of doing so is so that a large number of palisade cells can be packed per unit area, and therefore the amount of when light penetrates into the leaf, large amount of light, these are the main light capturing cells in the dicot leaf. So large amount of light can be captured that way than distributing them the cells in another way. So this is definitely another adaptation to maximize light capture. Option 3, distribution of veins throughout the leaf blade. Now it is not a direct benefit, it's not a direct modification, but it is also an adaptation to maximize light capture because the distribution of the veins spread throughout the leaf blade will actually enable the leaf blades to stay that wide open instead of folding or crinkling or withering. That will cause the surface area to decrease, right? So to prevent that from happening, the veins distributed throughout the leaf blade actually provides a sort of mechanical support. So that way it also enables maximum light capture. Most of y'all have selected this as the answer, and I will tell you why it's wrong. Option 4, leaf blades are oriented at right angles to incident light. Yes, this is also correct, and the benefit, now we know dicot leaves are definitely, in most cases, they are oriented right angles to incident light. Therefore, even at low light intensities, maximum amount of light can be harvested by the leaf. And option 5, increase surface area of leaf blade. Increase surface area, usually when the surface area of the leaf blade, this is where mainly photosynthesis takes place, the light capture also takes place, and therefore increased surface area will directly increase the light capture. Now why option 4 is the incorrect answer as cannot be considered is because, read the question again. I said, which are the following structural modifications of the dicot leaf? Structural modifications of the dicot leaf. If you check option 1, 2, 3 and 5, they are all modifications of the dicot leaf. But if you check option 4, leaf blade oriented at right angles to incident light, this is not a structural modification of the dicot leaf. This is a physiological adaptation of the plant body. The dicot plants, they tend to arrange the leaves at, keep them perpendicular to light. It's a physiological adaptation of the plant body, not a structural modification of the leaf. So that way you can consider option 4 as incorrect. That is all, and that is the only reason why this option is incorrect. Okay, question 13, allosteric regulators. Now you have to choose the correct statement in this. This one also most of y'all have got confused unnecessarily. Allosteric regulators are always activators. Now why do we say allosteric regulators? Because they may activate or they may inhibit that process. The reason we say allosteric regulators is they may be activators or inhibitors. Now these allosteric regulators, they play a very important role in the natural allosteric regulation of the enzyme activity in living cells usually. Now they are not always activators, there may be activators or inhibitors, so option 1 is incorrect. Then option 2, may bind to enzyme active sites. Now allosteric regulators, they always bind to a location elsewhere than the enzyme active site, and you call this location as the allosteric binding site. That means allosteric site, or you can say regulatory site. So it is not going to bind to the enzyme active site, so may bind to enzyme active site is incorrect. Third option are reversible non-competitive inhibitors. This is what most of y'all have selected as the answer again, and it is incorrect because allosteric regulators are not inhibitors. They may be activators or may be inhibitors. But the thing is, their function is like reversible non-competitive inhibitors. The resource book clearly mentions allosteric regulators act as reversible non-competitive inhibitors. The reason we're using this comparison is they come and they bind to a location elsewhere than the enzyme active site. Same thing as non-competitive inhibitors, and they are reversible. Non-competitive inhibitors, they may be reversible, right? Inhibitors, they are reversible and irreversible ones. And allosteric regulators, they don't always bind. When they want to stimulate an activity or inhibit an activity, that particular regulator will come and bind. So their function is like reversible non-competitive inhibitors. They are not reversible non-competitive inhibitors. Option 4 stabilizes only the active form of enzymes. This is wrong because allosteric regulators, they will regulate between two stabilized forms of enzymes. One is the active form they stabilize, that is done by the activators, while there's the inactivators or the inhibitors, they stabilize the inactive form. And since I said allosteric regulators, again option 4 is incorrect. And option 5 binds to enzymes having more than one active site. I have not said binds to enzymes always having more than one active site, and I said always binds like that. And resource book also clearly says most enzymes which are regulated by allosteric regulators have more than one active site. So that way the most appropriate and correct answer is option 5. Question 14, Which of the following is incorrect regarding phloem translocation in green plants? Why I say green plants is it may be the angiosperms, the gymnosperms, the vascular plants, you name it, anything. So we are going to check the incorrect statement regarding phloem translocation. First one takes place through living phloem tissue. Now most of you all will get confused that the phloem tissue, it has mainly living cells like the sieve tubes, the companion cell, the parenchyma, and what about the phloem fibers? Now fibers are sclerenchyma cells, so they are dead, right? Well, that's true, they are dead, but the phloem tissue is not, as a whole, it's not dead. It's a living conductive tissue. Therefore, statement 1 is correct actually. It takes place through living phloem tissue. And if you all are still going to be against my answer, this is actually discussed with tutors. Remember, all these answers are confirmed with senior experienced teachers. Now, another thing is, if you consider the phloem tissue, we say it has phloem fibers, right? Yeah, dead cells. But fibers are sclerenchyma, and sclerenchyma cells are dead cells at functional maturity. So in a young developing phloem tissue, which is also conductive, they are the cells which are still developing, not attained functional maturity yet, even though they are still functioning. At that time, the phloem tissue will be, the sclerenchyma cells may be still living. But the thing is, we say they at functional maturity. That means when they attain functionality. That means usually conduction doesn't occur also in such phloem tissues, but as a whole, it takes place through living phloem tissue is correct. Check statement 3. We'll come to option 2 next. Option 3 mainly conducts sugars through sieve cells. Now, there are no sieve cells in angiosperms. They have sieve tubes. Sieve cells is found in the gymnosperms and the other vascular plants. So, mainly conduct sugars through sieve cells is correct, because I said green plants as a common. And they mainly conduct sugars is also correct, because the 30% of the weight of the phloem sap is actually sucrose. So that way option 3 is correct. Companion cells, option 4. Companion cells function in moving products of photosynthesis from mesophyll cells to sieve tubes. Now the products of photosynthesis is what is going to be moved during phloem sap translocation actually. And yes, it is the companion cells. During phloem loading, one of their main functions is to move the products of photosynthesis from the mesophyll cells where it's produced to the sieve tubes, where it's going to be transported to the place where they're going to be consumed or where they're going to be stored. So option 4 is also correct. And 5 takes place due to a positive pressure push along sieve tubes. This is also correct, because phloem translocation, it involves, according to the pressure flow hypothesis, it involves a positive pressure push actually along sieve tubes. That's how translocation occurs. The neighboring xylem tissues, they have water at higher water potential, while the water potential in the sieve tubes is very low. That high solute potential is there. Therefore, water will diffuse and enter passively by osmosis into the sieve tubes. And now, where they diffuse and enter in, their water potential will rise a bit. But right in front of them, the solute concentration is again high. So that means the water potential in front of them is directly low. So from where they are, they want to go to always they want to go towards the lower water potential gradient. Always they want to go that way, along the gradient. So water will this way push, as it tries to move forward, it will push the entire phloem sap content. That's how the pressure flow hypothesis is explained. So that's also correct. Then the incorrect statement would be option 2, where we say phloem loading is an active process. Now it is also correct, because we are not saying it's always an active process, but since we say select the most appropriate answer, this is incorrect, because we'll say phloem loading involves movement of phloem sap from the mesophyll cells into the sieve tubes, right? Now this movement may be movement of the solutes along the apoplastic route, the transmembrane route, or the symplastic route. It may involve any three. But when it involves the symplastic route, it never involves any active process. Even though it may involve active process when going through transmembrane routes and all, it will never involve active process when it transposes through the symplastic route. So if it uses the symplastic route, phloem loading is also going to be continuously passive. The reason why I say it's an active process is because mostly it occurs also through the apoplastic and transmembrane routes also. Therefore, it is not an incorrect statement as a whole, but from here it is the most appropriate incorrect statement. So question 15, during photosynthesis. Again, you select the correct statement. Option A, chlorophyll A is the key light capturing pigment in all photosynthetic cells. This is incorrect because you study in microbiology the photosynthetic bacteria, purple sulfur bacteria and green sulfur bacteria. They have bacterial chlorophyll as their key light capturing pigment. They don't have chlorophyll A. So in all photosynthetic cells, this statement goes wrong. Chlorophyll A is found in most but not all as the key light capturing pigment. Option 2, carotenoids function in photoprotection. Consider plants. We have not said all, right? So we'll consider plants. In plants, there's special pair of chlorophyll A for its photoprotection. That means we'll talk about what it is. It is the function of the accessory pigments to do that. It's coming under the unit 2. One of the main functions of accessory pigments is to provide photoprotection to chlorophyll A. Now, accessory pigments includes chlorophyll B and carotenoids. So it's correct. And photoprotection actually refers to that absorption and dissipation of excessive light energy, because one thing, excessive light energy may damage the chlorophylls, or else they will react with oxygen to generate oxidative reactive molecules which are harmful for the cell. So that's why it's very important process. So that is the correct statement. Let's check the other three. ATP produced by oxidative phosphorylation is used up in reactions of Calvin cycle. This is incorrect because it is not ATP produced by oxidative phosphorylation. ATP is produced by photophosphorylation. That energy is only used in reactions of Calvin cycle. Okay, and photophosphorylation occurs when the linear electron flow at that location in the PS2. When electrons move from the primary electron acceptor PS2 to the low energy state, special pair of chlorophyll A in PS1. Question option 4, ribose bisphosphate is the carbon dioxide acceptor during carbon fixation of the Calvin cycle. I said read the question carefully. It is not ribose bisphosphate, it's ribulose bisphosphate. Ribose bisphosphate is an even more complex compound, so therefore this answer is also incorrect. It is ribulose bisphosphate which is the carbon dioxide acceptor during carbon fixation. And option 5, light dependent reaction occurs only in the thylakoid membrane system of grana, or grana you can say. This is also incorrect because I am saying during photosynthesis as a whole. That means it includes the cyanobacteria, all photosynthetic cells. Now in eukaryotes, yes, light dependent reactions occur only in the thylakoid membrane, but it's not only in thylakoid membrane. Now if you consider the prokaryotic cyanobacteria, light dependent reaction occurs in their plasma membrane. It forms inner foldings, and in that only it occurs. So it's not always in thylakoid membranes. They don't have grana. They form these inner foldings, and in that only there are photosynthetic pigments also present, chlorophyll A and phycocyanin in cyanobacteria. So that's how they take part in light dependent reaction. So again, most appropriate answer is option 2. Thank you very much for all the students who attempted this paper again, and you can trust us with the options again, because all questions are, like I said, prepared with the discussion of seniors and also the experienced tutors, and that way when we are making the questions also, another thing is, all these questions are our self-prepared questions which you can find in the study link papers, so you can use it as a revision before we come to the next quiz. And hope you all get benefited. Just learn to read the questions carefully and follow the instructions, and everything will go well. Till next time, have a good time. Have a great time, everyone.